I'm Lucy, and I am Czech and Polish.
I'm Danielle, and I'm German, Irish, Polish, and Welsh.
I'm George, and I'm Egyptian.
I'm Jack, and I'm Irish and German.
Many of us know where our ancestors are from based on stories that get passed on through the years. But it is also possible to know your family history based on patterns in your genes. There are different sections of your DNA that have repetitive sequences, and different populations tend to have different numbers of repetitions. By knowing the sequence of these sections of your DNA and comparing them to people in different regions of the world, it's possible to predict from where in the world your family has migrated throughout history. For example, the ALU element is a sequence of about 300 base pairs found in many locations throughout the human genome. Insertions of ALU are considered irreversible and happen infrequently. Researchers have been able to identify when and where many of the insertions first occurred and can use a person's insertion pattern to help determine their ancestry. In many instances, it takes quite a long time for one group to have a genetic pattern distinct from others. However, there are some special cases of migrations that can have drastic effects on an entire population, like migrating to a totally new place or not being able to leave a certain region. When a small group from one population moves to a new place where no one else in the population has been, a phenomenon called the founder's effect occurs. Since the new population is started by relatively few individuals, the alleles of the founders occur at a much higher frequency than they did in the original population. Instances of the founder's effect impact future generations, as they will have much higher frequencies of these founding alleles than the original population, changing the course of evolution. This happened with humans in 1658, when 344 people sailed from the Netherlands to South Africa and started a colony there. The Dutch group began to reproduce, and the colony grew from the mere 344 individuals that they started with. However, one of the founders, a man named Willem, had Huntington's disease. We mentioned earlier that a way genetics can be used to trace migrations is by comparing repetitive sequences of DNA. Many times these sequences did not fall within coding regions of the DNA and did not have physical effects on people. However, Huntington's is caused by repeats of a triplet, CAG, in the Huntington gene. Most people have between 0 and 35 of these repeats, but when the number reaches 36, a person is at risk for developing Huntington's disease. If it's above 40, the person is guaranteed to develop the disease. This causes the effects of Huntington's to proliferate, mainly the degeneration of the nervous system. This disease has a dominant inheritance pattern, so when Willem began to have many children with his wife, it's likely that about half of them also carried the disease. As his kids began to have kids themselves, they continued to pass on Huntington's. Since the population was so small to begin with, the proportion of offspring that came from Willem's family and likely had the disease was very high. Today, over 200 individuals from 50 different families of South Africa's Afrikaner population have been diagnosed with Huntington's. This translates to about 0.0074%, which seems small but is nearly 3 times greater than the occurrence worldwide. Since Huntington's is fatal, many individuals do not live to reproduce and pass on the genes for the disease. It's very likely that closer to Willem's time, the proportion of individuals with Huntington's was significantly greater than it is now. It's been hundreds of years, and at least 14 generations have passed, yet we can still see the effect that one founder had on a small colony as it grew to a much larger population. Another way that migration patterns can affect evolution is called the bottleneck effect. The bottleneck effect occurs when a population becomes drastically smaller, reducing genetic variability and changing allele frequencies. The bottleneck effect can occur as a result of an event isolating part of a population, for example, a flood or a fire separating some members of the same population. In this case, individuals cannot migrate and are limited in mates. Thus, the size of the gene pool for the new subpopulations is much smaller than that of the original larger population. Like with the founder's effect, the allele frequencies shift so that the alleles of the originally isolated individuals occur at a higher frequency than they did in the original population. Sometimes, entire alleles are lost, if no one in the new isolated population carries them. With the loss of some possible traits and increased representation from the original individuals, the genomes within the population become more and more similar as the individuals reproduce over time. An extreme example of this in nature is the Tasmanian devil. Tasmanian devils are marsupial carnivores that were originally native to Australia and are now found only on the island of Tasmania. Devils are found only in Tasmania because of three main factors driving them extinct on the mainland. The first is the introduction of Asian dogs, dingoes, which outcompeted devils for food. The second is increased aridity of the land, which led to less food for devils. The third is the fact that farmers mistakenly blamed devils for killing livestock and tried to eradicate them. Since these events reduced the population to a small size by limiting their habitat, and the individuals were unable to migrate off the island, there was a huge impact on allele frequencies, and thus the evolution of these Tasmanian devils. Over time, populations of Tasmanian devils have evolved to be so closely related that individuals can hardly be identified as unique, even when looking at the genomic sequence. They are very aggressive and interact largely through biting each other. Whether devils are competing for food or otherwise, these bites are aggressive enough that devils would wound each other very frequently. This biting is especially concerning because it facilitates the spread of devil facial tumor disease, or DFTD, a facial cancer that is transmitted between devils through biting. Normally, a tumor is not contagious between individuals, as the immune system recognizes cells from another individual as foreign and destroys the cells before a tumor can form. However, due to the bottleneck effect, the genomes of individual devils are almost identical. As a result, tumor cells from one individual are recognized as self by other devils, and the cells are protected from immune response. The tumor cells then proliferate and cause devil facial tumor disease in the individual that has been bitten. DFTD currently threatens to drive Tasmanian devils extinct. The disease has spread across the habitat to almost all populations. The lack of genetic diversity within and among populations not only makes the cancer transmissible, but decreases populations' ability to evolve towards resistance. Because there is so little genetic diversity, natural selection cannot select for those that have the traits to survive. For Tasmanian devils, the bottleneck effect has had a big enough impact to threaten extinction. The Tasmanian devil has been a protected species in Australia and Tasmania since 1941. But now that DFTD is such a large threat, significant conservation efforts are being made. Many populations of devils are almost genetically identical to each other, but there are some populations that have some significant genetic variants. Conservation efforts include introducing these populations to increase genetic diversity. The hope is that increasing genetic diversity will prevent transmission of DFTD, as well as allowing mixing of genomes and selection pressures to produce offspring that can more effectively fight DFTD when it does occur. These efforts have led to successful increases in genetic diversity and decreases in incidences of DFTD within the closely monitored experimental populations. However, this has not yet translated into the wild. The hope of these projects is to release the devils into the wild to increase genetic diversity and decrease DFTD, as well as possibly setting up completely new disease-free populations.